a quick overview of what we are doing right now with energy and matter. As you know, that right now we're doing an experiment looking at yeast's optimal niche in terms of cellular respiration as a function of temperature. So cellular respiration is a chemical reaction that occurs in all heterotrophs. And heterotrophs are organisms that undergo chemical reactions in which they break down and rearrange chemical bonds by consuming food rather than producing their own energy. So autotrophs, or photosynthetic organisms like plants, build their own carbon-based energy to use, while us humans and squirrels and squids and whales, anything that's a heterotroph needs to use glucose with oxygen to produce energy, usually in the form of ATP, which we'll talk about more in future classes. The reasons why these reactions happen is, one, it produces heat for body warmth, and depending on the individual species that we're looking at, they might need a lot or a little of body heat to stay alive in their environments. It produces usable forms of chemical energy, so sugar unto itself is too big and too reactive to actually use to do work within our cells. And it helps recycle matter back into the environment. If you know anything about photosynthesis, you know that CO2 and water are the inputs or the ingredients for photosynthesis. If an organism runs out of energy to react within their systems, they will die. So when we're thinking about any chemical reaction, there's two components to it. The first are the reactants, or the inputs, or the ingredients. These are the things that are used to make a reaction occur. In cellular respiration, we use one molecule of glucose with 6 molecules of oxygen. And the oxygen is a reactive molecule that helps split and cleave and rip apart the bonds in glucose. And our products are the molecules that are produced from rearranging chemical bonds. In cellular respiration, we make 6 molecules of CO2 and 6 molecules of H2O. These are considered waste products. And we also produce ATP, which is chemical energy. The other thing that we produce as a function of this reaction is heat. About 70% of the energy stored in glucose is actually not carried over to CO2, water, or energy from ATP, but rather it's lost as heat. It's another form of waste. So two things that we need to consider with any chemical reaction are, how does it show conservation of matter, and is it an exergonic or endergonic reaction? Any chemical reaction in any system, whether it's living or non-living, demonstrates the conservation of matter, which means that the number of atoms for each specific element, carbon, oxygen, etc., remains the same before and after the chemical reaction. Matter is not destroyed, it's just rearranged. So if we look at this reaction that's balanced, we have 6 carbons and we have 6 carbons. We have 12 hydrogens and we have 6 times 2 hydrogens. We have 6 plus 6 times 2, which is 18, 12 plus 6, 18 oxygens. And we have 6 times 2 plus 6 times 1, 18 oxygens. This is showing us the conservation of matter. So we are rearranging our chemical bonds, and we're not losing anything. And because of that, what we're losing or changing is energy, or the ability to do work. So the reactions that are being created are producing low energy products, which means that we have an exergonic reaction. Exergonic reactions occur when you take high energy reactants like glucose and make low energy products like CO2 and water vapor. They're called exergonic because the energy, exer, is being released into the external environment. So exer is external, gonic is energy or heat. We're releasing energy and heat out into the broader ecosystem. For us, that would be our bodies and then the air around us. If this was a wood stove at your house, it would be logs being burned to produce heat and light and sound to heat your house, and the products being ash. Energy is being released into the environment when we rearrange those bonds, and the products are less usable. We can't use CO2 or water to produce any form of energy in our bodies. They aren't able to produce work or energy for us. So our task with our first lab is to consider whether or not Saccharomyces cerevisiae, or baker's yeast, which is a single-celled heterotroph, it's a fungi, it's not a bacteria, undergoes aerobic respiration based on the function of temperature. We're going to be asking, does the temperature of water affect the rate of cellular respiration? We think, or we should assume, that there's an ideal average temperature that is the highest level of CO2. And as you move away from the average, whether hotter or colder water, we should see losses in efficiency. And this should allow us to see the fundamental niche of yeast. At a certain temperature, really cold or really hot, we should be killing off the yeast, and there should be no cellular respiration. Our independent variable for this lab is temperature. We are modifying the temperature of water, and our dependent variable is the amount of CO2 being produced by yeast. Every time you have so far used 50 milliliters of water, 2 grams of sucrose or table sugar, 1 gram of yeast, and 5 minutes of CO2 measurement. And we're really measuring the amount and rate of CO2 production. So I hope that was helpful for overviewing what cellular respiration is and what this lab is trying to attempt to do. Be prepared to answer a few questions about this tomorrow in class on a quiz when you walk in. I hope you have a good rest of your day. If you have any questions, please ask or see me.